what used to be a big puzzle. And the way that's done is you have DNA, which is a double helix, and it gets copied to a single strand of RNA, which also consists of 4 different bases. And now you have to go from here to the language of protein, which is a chain of amino acids. And the way that's done is groups of 3 bases are read to code for an amino acid. And Crick correctly predicted that there would be an adapter molecule, and that's called tRNA. And what tRNA has, this is simply a cartoon of tRNA, it has 3 bases that base pair with the triplet, with a group of 3 bases on mRNA, just like the bases base pair within the double helix. So it's the same kind of chemistry of base pairs that occurs in the double helix, but this time it's between tRNA and the messenger RNA moiety that carries the information. So you can see that a tRNA comes in, it recognizes its triplet in pairs and brings along its correct amino acid. And then the next tRNA comes in and it brings in its amino acid, which is different, or it could be the same, but potentially different. And then these 2 amino acids have to be joined together. And then the process has to continue along this genetic message. So essentially we're taking genetic information present in our genes, and using that information to make chains of amino acids, which are proteins, which fold up. Now, clearly, this doesn't happen by itself. And that was realized very early on when people who were looking at where newly made proteins were in the cell by labeling using radioactive amino acids and doing electron microscopy, they showed that newly made proteins localized on these particles in the endoplasmic reticulum. And these particles were isolated, and they contained, they all were about 250 angstroms in diameter and contained 2 subunits, a large subunit and a small subunit. And it turned out that all species of these particles from all species consist of a large and a small subunit. And these particles were called ribosomes. Now, most of you know Jim Watson because of his discovery with Francis Crick of the double helical structure of DNA. But it turns out that his lab actually pioneered some of the early work on ribosomes. But because he didn't actually do the work himself and didn't actually write the papers, but he allowed his students and postdocs to work very independently, he didn't put his name on any of the papers. Now there's a novel concept that perhaps, big, Indian scientists might take note of. So here he is talking about the early days of ribosome work. Oh, 1 minute.
A little round outfit doesn't contain DNA, but contains carbon. And I started studying it with Alfred Tischer, here at Harvard, and we knew that it was involved in on the ribosome. A little round outfit doesn't contain DNA, but contains carbon. And I started studying it with Alfred Tischer here at Harvard in spring of 1967. And we knew that it was involved in how proteins are made because the newly made proteins were tested. So we thought if we solved the structure of proteins of the ribosome, we would know how proteins. And that was a big puzzle because we had to somehow go from DNA to information, DNA to information proteins. And there was this particle, which we thought was round and circular. Hopefully only one sort of protein. If it did, then maybe it would not be easy to solve, but it would be solvable. So over the next couple of years, after I assembled some people, first the protein chemist who looked at it and said, I'm leaving. And because it wasn't simple. And then a protein chemist actually said, gee, there's 30 proteins for a lot of things. And so automatically I knew we'd never know its structure.
So it is true that in Watson's time, in the late 50s, it would have been impossible to determine the structure of the ribosome, but it didn't stop a large community of biochemists worldwide from characterizing the ribosome because it was so important. And they first figured out what the ribosome is made of. There are about 50 proteins and 3 pieces of RNA in bacteria, and then they showed how the subunits would assemble, and this is what an electron microscopy image of the ribosome looks like. And then about 40 years of biochemistry showed that the ribosome, the small subunit binds to the genetic message, and the ribosome has slots in it for these tRNA adapter molecules. And the way that happens is the first amino acid binds, and then the second amino acid binds in the second slot, and that enables a bond to be formed, which perhaps Tom will talk about in his talk. And so you get a bond between the first and second amino acid. And then the whole assembly has to move, and so that results in the first tRNA being ejected, and then the ribosome assembly is moved to the next triplet, and then the third tRNA comes in, and then a bond is formed, and so on. So you can see from this picture that the message, the information is being read here, while the polypeptide chain of amino acids is being synthesized over here, and that's done by this large, complicated machine called the ribosome. Now remember that my cartoon picture, this is a highly simplified picture of the ribosome, and the first breakthrough was in understanding what this tRNA molecule looks like. And actually it doesn't look like this, it's simply a conceptual cartoon. It looks like this, it's L-shaped, and here's the anticodon triplet that base pairs with the triplet. So this base pairing actually occurs here, and the amino acid is brought here. Okay? And this is the view of the ribosome we had in the kind of early 90s, where here's the small subunit, and it binds the message, presents the triplet, the L-shaped tRNA binds the codon here, and the amino acid is brought here to an active site where peptide bond is catalyzed, and the nascent peptide emerges through a tunnel in the ribosome. Now, this was, of course, tremendous progress from knowing nothing about the ribosome to knowing a lot about it. But if you want to understand the nature of the ribosome in chemical terms, and if you want to understand how antibiotics bind it, you need a high-resolution structure of the ribosome. And to do that, you have to understand how we normally visualize small objects and see detail in small objects. The way you do that is, if you have a small object and you want to look at it in detail, typically you would use a lens. And what a lens does is it collects scattered rays from the object and recombines them. And if you use your lens correctly, the image will be magnified, and then you can see that with your naked eye. Now, in fact, all of you are using lenses to make an image right now when you're seeing the screen, because the light scattered from the screen is going all over the auditorium, and you're taking a portion of that light, and you have a lens in your eye that recombines the rays and forms an image on your retina. And that's why you're able to see this image. However, there's a law of physics that says that you cannot distinguish detail that's smaller than the wavelength of the light that you're using. And the problem is that light has a wavelength of about 5,000 angstroms. And the distance between atoms in a molecule is typically about 2 angstroms. And so you cannot using light, ordinary light, use a magnifying glass or a microscope in order to discern details of the atomic level. You can use a technique called, you can use light of a much shorter wavelength, and if you use light of the appropriate wavelength, which is 1 to 2 angstroms, those are X-rays, okay? So X-rays are simply photons, just like light, but they're much shorter wavelength. The problem is that there's no lens that will actually recombine the rays like an optical lens. There are lenses developing now, but they don't have sufficient, they're not there where you can look at individual molecules. The second problem is that X-rays are damaging. So if you look at a single molecule, just like you would look at an object using a magnifying glass, the amount of X-rays you would need to get enough signal from a single molecule would actually destroy the molecule. So even if you had a very powerful lens, you wouldn't be able to get, using continuous X-rays anyway, an image of the object. But there is a technique that you can use to circumvent that, and that's called X-ray crystallography. And in X-ray crystallography, what you do is you crystallize your object, which means you form very regular three-dimensional arrays of the same object, like the ribosome. And you take that object and you put it in an X-ray beam, and you hit it with X-rays, and the X-rays are scattered, just like any light is scattered from an object that it hits. Okay, so X-rays are scattered, but because it's a crystal, it has two consequences. Unlike light rays, which are scattered in all different directions when they hit an object, when you have a crystal, the rays are only scattered along certain directions because of only those directions have what's called constructive interference. The other thing that happens is that the signal from all the millions of molecules in the crystal is added so that you get enough signal even from these macromolecules, which are small by our standards, of everyday standards. And so you get enough signal to measure the scattered rays. And what these scattered rays look like is like this. This is an X-ray image, a detector image, and you can see the scattered rays are in these spots along the detector. And what the process does is it takes these scattered rays and recombines them into an image, and that produces a three-dimensional image of the object. Remember, whether there's a lens there or not, the scattered rays are present from an object. So even if you are not sitting in your chair, the scattered rays from the screen or beam would actually hit you, hit your chair. And the fact is that you have a lens that collects the scattered rays and makes an image. So you can do in a computer what a lens does in real space, which is to collect these scattered rays and recombine them to make a three-dimensional image. Now I'm simplifying the problem because there is a problem called the phase problem, which I won't go into. But that's fundamentally what we do. We recombine scattered rays in a computer to produce an image. Now, in order to do that for the ribosome, you need crystals of ribosome, and the first breakthrough was made by Ara Yonah, who was here yesterday and produced crystals of the 50S, and then a group in Russia produced crystals of the 30S in the entire ribosome, and then a number of us had to contribute in order to eventually get a structure. Now, one of the ways that, one of the things we needed